our last session of discussions. If you came in late, please stay at the end of the meeting. We're going to take a quick group picture. Please do not leave. Otherwise, I'll pass it on to the co-chairs.
Great. So I think we thought we would just go around the teams in whatever format you want. If you want to just share your thoughts, if you want to do anything up front here, we can do it that way. Verbally, I think, because we have the Zoom participants as well. Then we're just going to transition sort of when it's natural from this to the closing bits around the conference. We do have some questions we want to pose to everybody about that closing bit too. So would any of the teams, we can go down maybe just in the order here.
Yes.
Would team A like to start with their discussion?
Sure. Yeah, I guess I'll just kick off with some of the notes and thoughts from that discussion. Basically, we discussed a lot about bringing these orthogonal systems into C. elegans. So a lot of the genetic engineering circuits have not been built in worms, and that's a really large open field. And so, for example, we don't have a lot of inducible motors with small drugs and things like that. So we talked about, could we bring over some of these estrogen hormone receptors to do inducible gene activations, and then do that in a tissue specific fashion and build up those the raw materials that are needed to build these circuits. Discuss what kinds would be the most efficient circuits to do, and this would be one of the very first things we'd like to bring over into this animal system that already exists in a lot of these multiple dimensions. Then applying this to some of the more relevant aging pathways, DAPs, that.
It's really cool to learn what components of a synthetic circuit are already present or available in the systems, just that they haven't been.
Yeah, so we got very pragmatic very quickly about what could actually be accomplished in the next, almost in the next year.
Yeah,
That's excellent.
That kind of time scale.
Super nice. That's great. From a concept to maybe some actionable possibility, right? That's great.
And I mean, outside of aging, I think that there's
Right. Oh, yeah.
Elegance that
All right.
I wasn't even thinking about it. And I think I've thought about it all in my entire career either. That was a really pleasant discovery.
Right. Well, I mean, C. elegans, right? Like, I mean, Gordon Liskov, for example, we have all these like AD models of in C. elegans. They are like human disease genes that are for AD that are tau, A beta that are in C. elegans, and I'm sure for many other diseases too, right? So these tools could be used for many things down the road.
Yeah, not to drill too much into the the weeds, but why mechanically or otherwise has this not been accomplished sooner? That's the.
I mean, I've always had my perspective on the C. elegans field. So it's driven like a lot of important discovery, like RNAi and some of the first kind of cloning by transformation. But the field of C. elegans is about 1/10 the size of fly genetics, for instance, I think. And I don't know how it compares to yeast at this point. So what tends to happen in the field is when something works, everybody just does that until someone comes up with some new idea. So the analogy that I used is the field is given a hammer. Boom, 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 boom. There's not a lot of people inventing new hammers for worms, and whereas in other more competitive fields or more broadly applied, people are just hammer makers, and that's what they work on. So that's always been my perspective of the field. When something works, you just keep doing it until it doesn't work, and then. So, for instance, in CRISPR, worm people were very early adopters of making CRISPR work, but people inventing new CRISPR technologies are not working within worms. Instead, they kind of do stuff, and then the worm move it and say, oh, this works, or we can make this better. But people are not inventing new CRISPRs or CRISPR activation using worms as the model. So I think that's just kind of a field orientation of the kind of people who are working there.
it could, I asked because there is an analogy, and I think we're all lemmings, basically. I don't think
Yeah.
it's special for the C. elegans. Maybe that's not the word you use, but as
Yeah.
far as like, it's much easier to use something that definitely works, right? 'Cause manage the risk of trying to build your own thing for maybe no benefit. Until recently, that was also true in a lot of mammalian cells. And I think what drove a lot of these technological innovations was kind of what we're talking about here. Like, you can get by with PTA, you can get by with Gal4, like you do a lot with those until you can't. And then we had to like build a bunch more stuff and a number of different parallel, I would say yeast and mammalian cells both. There's been this proliferation in the past 5 years, things for doing more than what those foundational tools enabled, but it was only because we then started having the need for these more complex things to do the science. So it might be, this is a wonderful marriage of like the tech development and the driver for it.
Yeah, it feels like it's a moment in time kind of thing, and so it'd be good. The other term of art that we've definitely learned is the orthogonal approaches. So I think worm people have not been so concerned about the value of having orthogonal systems that don't kind of have all these mass pleiotropic effects and things like that, but it's bringing in the system that we were talking about is, this estrogen activation transcription factor. It's like, well, worms don't have estrogen, so that's
Right.
That's cool. Why doesn't that exist already? No one's done it or needed it or whatever, but has a potential for creating, more ways of manipulating. And that's why I think doing this on the road to aging is a good broad perspective, but anything that is accomplished as we do that will benefit all the genetics. So that's kind of a good double whammy, I think.
Cool. Just gonna say there's a neat analogy with plant synthetic biology too, where for a long while the gap was thought to be that, like, mature plants take a long while to grow, even Arabidopsis, but it's more than that. But like biochemistry is adequately different, that it took like a good amount of banging on it to figure out how to get some of those same concepts to map onto the new system. But yeah, it's cool. Neat. Okay, well, shall we move on to the next topic? We can always circle back to connecting points.
Yeah,
Yeah.
sure.
I wonder, can I ask one question there? I'm just curious, because there's, C. elegans seems like such a good system, and you mentioned flies and Drosophila. Are they as genetically tractable enough to also do these kind of things? These synbio circuits in vivo?
Yeah, there might actually, we haven't Googled synthetic biology of flies. I bet
Yeah.
there is some already being done. I just don't know.
There's some. There's a colleague of ours who builds these circuits for like monitoring and perturbing temperature regulation response to things like that. The lesson there when we were doing this kind of comparative, like where can we do symbio, was like his take was that flies were way faster than I had thought. Like that was years ago, and it was like $200 to make a transgenic fly version of whatever the thing is you're thinking about.
Mhm.
And then the biochemistry was adequately.
Part of the theme that we build around our own work is that we are at this transition point in worms, and probably people will figure it out in flies and other things, is that we're trying to make worms like a microbe, in that right now you can transform microbes by having DNA libraries, things like that, and pushing them in through the membrane. Can't do that with animals because of what's called the Weissman barrier, germline soma barrier. It just doesn't work. And so we're creating libraries that are already kind of preloaded into the germline,
Right.
and then we activate like a little surprise bomb in there. And so that hasn't existed before. And so we think that will work for these applications, but it allows us to build the preload all the parts and then kind of do stuff all at once rather than very preciously. I think that genetic transformation technology functions and all that stuff, it's been more laborious than working in microbial systems, even though it's only $200 for one transformation. What if you need 5, 10,000? Then here you are, 10,000 is crazy. So yeah.
All right, I think we could move on to study number B. Do you maybe like to start?
Get started. So we talked about the kind of challenges and opportunities of building sentinel cells that would record and potentially respond to markers of aging. So we talked both about cells that would age themselves, and so could be used to study the aging process and different markers of aging, as well as the cells that are really robust, healthy, and detect the marker of aging in context of a tissue or some sort of culture. Then we talked about different things that these sentinel cells could report on. So of course, there's a lot of data on transcriptomics and proteomics. We talked about molecules. There's a panel of molecules that senescent cells secrete. And so one of the challenges is identifying good markers of aging. So a lot of genes, proteins, or secreted molecules are kind of like kind of play profit effect or general markers of inflammation, for example. So you would need something that is a specific marker for aging.
Yeah, the EV thing, we talked about using synthetic biology as a way of kind of interrogating the sender receiver phenomena that involve the transmission of senescence and other aging phenomena by the secretome. And like, so like many things we know, like what the cells are spitting out, but we don't know like where it goes or what cells are impacted by that. So we talked about some specific systems one could build so that there is a sort of orthogonal, again, way of tracking when a signal is transmitted from a sender receiver. You can read that out by imaging or other modalities like that.
Yeah.
The list of candidate genes and transcripts and stuff we talked about.
Yeah, a lot.
Right,
Yeah, that's true.
right, exactly. Yeah, and lastly, I think we also talked about the specific tissue, the lung, for which we have particularly many data. And Dr. Gomez also mentioned he has access to like big transcript data sets. So we were thinking of maybe picking things that change on all kinds of levels, protein, transcript, secretome, and then maybe using, kind of prioritizing a little bit that way for first sets of interesting candidates. Yeah, so I think that was quite productive. So yeah. Kind of similar to yours, Patrick, where we went to like maybe some real action items, which is actually cool. And, this is actually, I was particularly happy about that. Right.
There was actually a fun thing that came up. This may be old news, aging phenomena, but one thing you said that was really interesting to me, I think to us, was the idea that you can identify these senescence hotspots in tissues from patients, and that there's a certain like radius of influence that and the mechanism by which that is spread is like not entirely known.
Yeah, that's actually one of the fun phenomena, and they call this also bystander senescence, right? And like, but I mean, it stops at some point, right? And that has to, right, as the whole body would become affected. So yeah, no, that's a really interesting. Right. Okay, cool.
Okay, let's go with the team C.
Okay, all right, so that was our team. So I'm going to go back and forth between different notes here. Okay, so our general theme, just to kind of recap, was sort of thinking of strategies focused on developing in vitro, sort of what we were envisioning were cell culture, right, based platforms. So assuming human cell culture platforms to understand aging at the single cell level. Right, so these would be sort of built-in, fusion proteins and gene circuits, so that we could sort of further interrogate what's actually happening within the single cell. So, just some general goals, right? Create a model system with engineered or, so eventually we want to apply this for hopefully in being able to induce aged cell states. And I use the quotes, I'll explain why in a second. And then so also, and as I mentioned before, to determine whether, drugs that were being screened as a therapy or drugs that, had some unexpected effects, how that actually affects aging at the cell level, right? So sort of like a drug testing platform. And then we also noted that, a sort of a pretty important need, right, that would complement what we're trying to do with synthetic reporters, right, that could give us a readout of whether or not the cell was in the age state. But we didn't get too deep into that because we knew that another team may have been covering sort of developing that type of technology. Okay, so we started our discussion by, just acknowledging the overall problem that there seemed to be no clear molecular definition of an age cell at the molecular level. But we put our thinking hats on and sort of thought of a way to develop a, sort of projects and research programs that would kind of work through that, that problem. And then so what we did was that, so I encouraged the team to think about, okay, let's go ahead and just list all of these, I guess sort of age-related cellular features that have been identified through correlative studies, right? So without really understanding whether it's a passenger or a driver, what studies have shown a correlation between X cell feature and age, compared to young cell. So we sort of built a short list of some things that really stood out in the literature. So these epigenetic clock, right, related features, DNA methylation in certain loci across the genome have been sort of regarded as a signature for an age state. And then, so that's a measurable feature. And then also a new thing that I learned was that just the sort of overall bulk integrity of constitutive heterochromatin. So for the non-chromatin people, this is the compacted DNA that tends to accumulate around the centromeres of the chromosome. So you have that, the core in the middle, and then sort of repetitive sequences on the outside, and all of that gets packaged really tightly into what's called constitutive heterochromatin. So apparently in age cells that loses its integrity, and I thought that was really fascinating, and that's definitely something targetable. And then whether that's a driver or just a consequence is sort of up for debate. Also other specific features, mTOR signaling, right? So that's something that can be targeted and manipulated. And then P16 expression, right, is this sort of a signature, a mark that's associated with senescence in the tissue setting. And so we had a really good discussion around being careful to prioritize markers that appear in a more physiologically relevant in vitro system, and trying to avoid things that might be artifacts of 2D culture. And so also we had a quick discussion about certain genes so far, what genes have been implicated in affecting aging. And so FOXO3 came up in ApoE4, right? So again, as long as you have a specific gene and you have a question about whether it drives anything, you can put that under, synthetic regulation and then test that idea. Okay, so starting with that list, then we went on to propose ways to use tools that are available in Synthetic Biology Toolbox to artificially control these features in meaningful ways that could actually produce, new knowledge. And then with the overall goal of determining, what role they have in aging. Are they drivers? Or are they, not sufficient to actually induce an age state? And then so we talked really quickly about this paper from Yang et al. 2023, this ICE mice system. And so I guess sort of the most important thing to note there, and I kind of emphasize this, is that, okay, regardless of what you think about, the results and their conclusions, the important thing that I think that they did was that this is a good example of doing something different to induce the age state. So they used precision targeting of, I think, 20 total sites in the genome, made cuts with sticky overhangs so that the DNA could just anneal properly, not produce any mutations, but then trigger this double strand break, sort of emergency repair cascade. So that right there, right, is a good example of somebody, doing something different to try to induce aging that's not irradiation or, chemotherapy or anything like that. Okay, so sort of taking things from there and sort of coming up with other sort of like precision targeting things we can do with synthetic biology, epigenome editing, right? So one of my favorites. If you're observing like methylation or some other, or histone modification showing up at certain genes in the age cells, artificially write it, right? Send in some epigenome editors, produce that chromatin remodeling, and see how the cell responds to that. And then also one that I had just learned about, right, the mTOR signaling pathway. And so that one got me really excited because, if we're talking about, protein-protein interactions out in the cytoplasm, what is it, Basher et al., I think was the paper, Wendell Lim's group, where they manipulated the mating signaling, right? So the sort of alpha factor, response pathway, use some protein-protein interaction, sort of artificial dimerization tricks to alter the dynamics of that cascade. So I think there's an opportunity there to apply that same approach in the context of aging, which I think would be really cool. And then so, degradation in, so a third target that we had, sort of identified in our list was this degradation of heterochromatin just sort of in general. And so, of course, right, sort of plugging approaches from our lab, we could build a fusion protein that would go in and find residual marks and then restore assembly of heterochromatin in those regions. And I believe that that's something that we could actually accomplish with techniques that we already have. And then so some other ideas that are just sort of more general and less focused on specific pathways. Yeah, we just discussed in general. So the advantages, and I think this is an important point to underscore, is that with synthetic biology, just always remember that what sort of makes what this field has developed really stand apart from sort of a typical, overexpression, of xcDNA, right, approach, is that we can build promoters that, right, respond to different inputs. We can control the timing and the magnitude of, whatever manipulation it is. And so we could take advantage of that to get a deeper understanding of how the dynamics of how changes in these factors that are implicated as drivers of aging might affect things. So things that you would otherwise miss by doing a simple overexpression or a knockdown, you could catch by doing, sort of timed expression, pulsed expression, and really playing around with the dynamics of manipulation. And then finally, the last sort of super important general point that we discussed was that it's very important to make sure that we're developing these in 3D culture systems, because you do see one of the participants, I'm sorry, I forget who was in the room, but had mentioned, oh, there she is right there. I had mentioned that there's a ton of artifacts, right, that keep showing up in the 2D culture versus 3D. And it's, we really ought to be careful to make sure that whatever platform we're developing is sort of compatible with a 3D culture system rather than 2D.
Cool.
Okay.
Well, thank you. That was very productive. Excellent. Wow, I love that. I mean, lots of really very good ideas already, and like also a little bit outside the box, which is cool.
Yeah.
So yeah, no, very good. Great.
Could I pull on one thread, and maybe Caleb, I might throw a question to you, since I think you know the story behind the paper, right? So Carmela, you made mention of this really interesting bit. I'm not sure everyone's familiar with this story. And here's the question. Some of these initial couple papers coming, including yours and the signaling pathway in yeast, and later working on TCR signaling pathways. My understanding from reading and hearing kind of Wendell's presentation of those stories is that those actually were not like hypothesis driven explorations. It was sort of a scramble, the connectivity, look at the resulting consequences thereof, and then that examination from that approach gave rise to all these kind of like interesting insights of what gives, and I think mainly of the T cell dynamics one in that later light. Is that kind of similar to what happened with the alpha factor response, or was it more a different approach?
I mean, I think the basic quantitative features of the mating response were kind of our,
Okay.
and we selected that because it was a good substrate for engineering, because the response was very vanilla. So we could, when we installed these feedback loops, which were based on protein interactions, so that was somewhat hypothesis.
Okay, cool.
So we're going to put in negative feedback. Okay, so we're going to see, a pulse and then attenuation. And,
Okay.
really the point of that was to show that just dumb recruitment between two factors that have different activities were going to be something that would allow you to predictably rewire the pathway.
Cool.
So engineering in that mode in mammalian cells is not really taken off.
Wait, there's only one example, right, to your point. I mean, there is one that I'm aware of. I mean, the mechanics are definitely different. So the delta notch, right, is a favorite, right, for these like signaling cascade, but that's the only example I can think of for mammalian
so
cells.
there's a really nice example, one example, and it even has therapeutic direction to it, is this, I can't, maybe somebody jump in, this Michael Lynn's lab put this out. It's basically rewiring EGFR responses in it's a synthetic recruitment trick, but in this case, it's phosphorylation responsive. So I mean, the tools are there to do that.
Yeah.
I think that maybe we need to know a little bit more about how mTOR works, but I think that it's absolutely good stuff to him.
Yeah.
I was asking that in part to bring it back to this, like, there's another biochemical mechanism we can use to interrogate some of these things. And also just kind of the like science of science part of that particular approach, right? Of like, how do we, in historical terms, solve biological, unopened or unanswered biological questions using these kind of approaches. That was super big. Neat. Okay. So shall we move on to team D?
So, yeah, so team D, we were focused on, payload delivery and targeting specific cell types, right? And please, you get, feel free to jump in at any point here. But yeah, so we kind of discussed initially what are the different methods that we can employ to do this payload delivery. And so, there are different options, like the viral or exosome or CAR NK cell, CAR T cells, right? These are all things we use. And then in particular, Mitchell has in his lab the platelet delivery too, which is an interesting approach. So of course, in aging, we're thinking in terms of what are the things we can target. And of course, one of the things we know we can target and to kill to help with aging is senescent cells. And so we can, utilize what we already know about senescent cells, which is potentially their cell surface markers, or we can use, some unbiased omic approaches or something to identify new markers, right? And do this in a cell type specific way, because we know that senescent cells are very heterogeneous, depending on tissue and whatnot. So it's nice in this case to, target something maybe specifically to one type of senescent cells. So, we discussed, using something like this platelet system to deliver a drug, senolytic drug, to kill senescent cells in a tissue. And also what's nice, right, about the platelet system is that they kind of naturally, you mentioned they naturally go to sites of injury or inflammation, which for senescence might be a good thing because they're often found at sites of injury and inflammation. We also talked about, different things that we can target, besides just the cell surface. You want to have specificity. So maybe we could, use one method is to first target the inflamed or fibrotic microenvironment. Like, so people do in the cancer field, they target the microenvironment. And then once the cell is there, then you also have the cell type killing mechanism that you can use as well. So using a combination of these two things. The other thing is, maybe there's something besides senescence that we would want to target. So is there, just the fibrotic, if there's damaged ECM, we know that ECM changes happen during aging. Can we detect and deliver something to the ECM to repair it? Yeah, that would be another potential target. Our conversation kind of went off to a little bit into, we started to think about sentinel cells as well, because, one of the things that happens in aging, everybody talks about inflammation, like inflammation is generally increasing in aging, but can we have some kind of reporter or quantification of that? So, maybe if you could have a cell that hones to sites of inflammation and you can kind of report it, and over the lifetime of an organism, where does this inflammation occur? How much does it occur? We get a precise readout of what is the inflammaging? Can we even get precise to, what is the source of the inflammation? Like it be homing to DAMPs, which are more from damage to cells, right? An injury type inflammation versus TAMPs, which is more bacteria or pathogen, right? So then even being more precise with what kind of inflammaging that we're quantifying. What was the other thing that we were, and maybe something also similar for fibrosis as well. We could do kind of reporter quantification for that. Another interesting reporter idea that we had was, we think about this idea that there are good senescent cells and there are bad senescent cells. So what is one way to distinguish these? And, one idea in the field is that senescent cells, they're meant to be transient, right? They're supposed to, if you have an injury or some kind of stress, then the cells become senescent, they kind of do their job to help resolve the wound, and then they go away, but for some reason during aging, they don't. So if we could now use some tools to develop markers for maybe early versus late senescence,
Mhm.
right? And then quantify those, and then if we could have, based on that, create a reporter for early senescent cells versus late senescent cells and do that in vivo, wouldn't it be cool if we could actually look at, you know, whether this is true in vivo? Do we see the accumulation of the late senescent cells, and that's what's causing, you know, likely driving aging related processes?
I actually really like that idea.
Oh, okay, yeah. So maybe there's a name for a grant, so.
Well, because I mean, I think that is always one of the problems. Not all senescent cells are detrimental and we all need them. So differentiating them and, you know, yeah, I mean, the good senescent cells should be temporal, right? And like, how did you call it? What was your word?
Transient.
Transient, yes, thank you. So, I mean, you and I have often discussed the temporal kind of aspect of senescent cells, and in general, I feel anything longitudinal in any the human or model system is always very insightful, right? Because monitoring over time, there's a lot of dynamic kind of changes that are there. So understanding those better and using sentinels for that, or developing sentinels for that, might be really interesting.
Yes.
I mean, that would be cool. Yeah. Right.
This derails, please stop it. Do we have an idea of if the loss of clearance of this bad senescent cells is due to lack of replacement or lack of clearance? Like, I'm thinking of when a kid loses their tooth, you don't get the wiggly tooth until the new tooth is dropping. It's by replacement that the tooth is removed. Do we know if there's a mechanism there that's broken that?
Yes, it's likely both. So as you age, you have more of the senescence inducing stresses in general, because it's kind of a more stressed environment that induced the senescent cells in the first place. But also we know there's immune decline with aging. So normally often the immune cells will kill the senescent cells when they're ready to go, but your immune system declines as you age, and it gets I guess worse at removing them. It could be due to the immune cells themselves, or it could be even the senescent cells somehow. When you're aged and a cell becomes senescent, it's not expressing the right, you know, cell surface marker or something for the immune system to see it, recognize it, and remove it. So it's, I think, both at both ends.
What about extrusion? Does that not have, do senescent cells just get, can they potentially get extruded? You know how they just kind of pinch off and then they go away? Or is it that an immune cells comes in a whole tissue and launches an attack? You know, do you know the difference?
Yeah, I don't know. Yeah, I don't know.
Loss of cell contacts.
Huh?
If there were loss of cell contacts or something like
Yeah,
that.
yeah, and it's like this way that the tissue can just like squeeze out and extrude a cell from a tissue.
That's cool.
So I wonder if there's like a mechanism that's gone awry to get rid of a cell that the tissue's on it.
That's interesting.
I don't think, as far as I know, nobody has looked at that specific.
I think so. Jody Rosenblatt
senescence.
in Cambridge is looking at extrusion. I don't know if she's applied it to aging specifically, but I was on one of her students' PhD committees. That's the only reason I know.
Okay.
So, but yeah, they do a lot of these really cool extrusion. Looking at this sort of mechanism. I don't know if she's applied it to aging though.
That's interesting.
Yeah.
I like that.
Can I pose a kind of follow-up question to the, mainly, I guess, the aging folks here related to one of the ideas that you mentioned? There are these senolytic CAR T cell therapies that exist, and they target UPAR for, I just looking up this one I had heard of from Michelle Settle's team. Is one of the challenges with, and there's in a model, some phenotypic response to this kind of treatment that's positive. My thought is that that's a very conserved marker of senescence, right? So would that be problematic because you target like all too many senescent cells? Is there an opportunity to like try to make these smarter in a sense, where, you know, making CAR T cells smarter is well within the wheelhouse of stuff that's been done? So that, is there a connection with the senolytic applications
I mean, I
So,
think
for the gap
that is one
yes.
of the key things, right? To pick something that's really very specific, right?
Yeah,
Because I mean, even markers like people like a lot P16, other cells express those too, right?
Right.
So I mean, I think that has been a bit the challenge, being really specific enough.
Yes,
Okay.
I think that even
Mhm.
UPAR probably is not that
Right.
specific, but
Right.
maybe in the certain applications it may work better.
Right.
Yeah, and there's like DPP4, but yeah,
Oh,
there's
yeah.
other cells that express these often, so I think yeah, it'll be important to get more precise
Mhm.
and more selective somehow.
Mhm.
And also, killing the bad and not causing negative side effects by killing the good effect of, good
Mhm,
functioning
right.
senescent cells. So
Right.
be something have to keep in mind when doing these therapies. Yeah.
Yeah.
for sure.
Yeah.
One of the reasons I was asking is, as been mentioned several times, one thing that we kind of can do as a field is control when and how and whether, under what conditions stuff happens. And so just knowing, like, we know this is a limitation, we don't know if it's a limitation or whatever around that could be helpful. Like, for
Right.
example, it might be that you only target UPAR, but you restrict activation to the tissue where
Right.
you know it's of
Yeah.
relevance and not in
Right.
tissue
Yeah.
where you know
Right.
it's of
Right.
harm, right?
Right.
Right, yeah.
Yeah, as said earlier, the problem is the systemic application of something. So if one could bring in a localization aspect in addition,
Right.
that would definitely help, right? So
Those are the two things. You either make it go where you want, or you
Right.
just let it go everywhere and control what it does
Right.
in the different places.
Mhm,
Yeah.
right, yeah, interesting. Mhm. I mean, going different places so far, the only way that was controlled is by localized injection, right? But if one had better molecular ways to direct where to go exactly,
Yeah.
that would be preferred, right? I mean,
Yeah.
because that could reach areas that you couldn't reach otherwise, right?
The fibrosis axis is another promising thing, because there are all kinds of things I'm guessing one could use to take that cue and think about what sort of like chemokine profiles and combinations and exclusions might be adapted to like a home to the, senolytic microenvironment,
Yeah.
if that's a thing.
Even if it's systemic, I don't know the answer. Could you, it was in a pause button, wipe it all, pause it,
Mhm.
see what comes
Uh-huh.
back.
Yes, that's a very good
Hope
point.
that your good stuff comes back and not your bad stuff.
Yeah,
That's right. I mean, you can use transient versions of any of these cell therapies
Yeah.
too, that would just
Mhm.
automatically have a limited transient phase.
and that's nice. I think senescent cells will be kind of amenable to the short or intermittent treatments, because it's like they reach it's a threshold effect. So when senescence burden gets high enough, then it becomes, systemically, and they persist long enough, then it's detrimental. So really, you don't have to eliminate them all. You just, maybe you'll pulse the senolytic, get it back down to the below the threshold, the pathogenic threshold. And hopefully you have minimal side effects when you do it. And, it's nice with senescence is that you don't have to have a perfect thing. Like with the chemotherapy, you want to eliminate every single cancer cell. But with senescence, you can just, kill a lot of them, and they don't, just replicate
Right.
out of control. So it's okay if you leave. So, yeah.
That is actually something that Judy Campisi has also discussed. Maybe one doesn't have to be like, get rid of everything, but just get rid of enough so the body is sustainable again, right? Like, so
Yeah.
that is actually an interesting thought. Versus in cancer, yeah, I mean, the aim is really always getting everything gone. Totally, right? And
It's the interesting thing, because that is generally, but that's not the only philosophy maintaining health in cancer,
Mhm.
right? And so I was gonna ask, when I first heard about these like senolytic CAR T cells, I had that idea, like get rid of the senolytic cells. We spent the last like 2 days talking about how inflammation is one of the key drivers of senescence.
Right.
Nothing sets up inflammation like a bajillion CAR T cells going off at the same time, right? So
Right.
I was wondering if that is kind of a challenge within general, that approach to doing senescence removal, and whether other senescence targeting drugs do consciously avoid or manage the immune consequences that are adverse.
Right.
Yeah, that's a good question.
Mhm.
I don't have experience with like immunotherapy, so I
Yeah.
don't know, but that's I think a very good concern to have.
Just
Yeah.
thinking about it, because normally when you're going after a cancer with CAR T cell, there are these whole like, if the acronym game in CAR T cell space is the top. type of science, but like armored cars are cars in which you like deliver immune potentiating factors
Mhm.
along with the CAR T cells,
Right.
right?
Mhm.
So hypothetically, you could do a different thing here, which is like a senolytic T cell with like immune modulating factors, immune suppressing factors, where you like find a balance where you get the thing you want, but you maybe like, maybe you don't suppress the CAR T cell itself, but you suppress like all the macrophages, stromal cells and things like that that are at the site where that T cell is going to go off.
Interesting.
Right? For-
Yeah.
it would limit some effect, but it would maybe be a balance.
Solves that.
What's that?
So your senescent cells are also good in pro-inflammatory.
That's true, yeah.
So, I mean, I'm not sure in CAR-T what causes your cytokine storm, but if you're removing something that does promote inflammation, which is your senescent cells, are you really have that side effect?
That's a good point. You mean like in the acute phase, right?
Yeah.
It's a very acute phenomenon.
You
Yeah,
do want your pro-inflammatory macrophages to be able to protect cells.
yeah,
You just
Yeah.
don't want to stay in that.
Right.
inflammatory state.
Mhm. Mhm. Cool.
Cool.
Right. Well, it looks like all 4 breakout groups had a great time and good discussions. That's amazing. And it was fun to hear. I mean, a lot of actionable ideas, I feel. Yeah, so I think like that's great. Thank you everybody for being so active. I mean, you're really making the success of the workshop, right? By being active and contributing and thinking and right, Josh, I think we had a really nice time yesterday seeing the discussions and it continued into today. I actually thought it was nice having a little bit the evening in between where it could settle in the brain a little bit and digest all these very novel ideas I think we've discussed throughout the day. Yeah, so I think some of the small groups had already discussed a little bit to kind of follow up a little bit by email ideas developed together. So I would actually encourage that now that you know each other. And because I do think that there are some really cool aspects that potentially could lead to some experiments or, you know, applications. So I think we definitely will follow up in our last breakout group. I hope others may too. Yeah, so Josh.
Yeah, I guess we had a couple, we'd maybe just transition to some of the stuff we wanted to do. We can, is it cool if we transition to sort of the wrap up portion of this?
Yeah.
Okay, cool.
Right.
So one of the things we wanted to do was take a moment and just ask if anyone had any key like surprises or learnings that were new. And this can be trainee or PIs, no one way or the other on this. So any like really interesting things you didn't know before and that you know now that are maybe useful. I'll mention like Kayla's mentioning of the opportunity to apply things to worms. It's one of those things that for 20 years is like one of those connections that occur.
Yeah. Well, actually, this was something we discussed briefly yesterday evening because I was recently at some dinner where we also had like a whole day meeting with a group of many people, and we asked what was the most interesting thing you heard. And it was so interesting to hear from the different people because people had really different things. So we just thought if you just think for a second what and just say what you really think. Yeah, AJ, you raised your hand. That was maybe just
Personally, I learned a ton about the juxtaposition between what happens in aging versus all of the other disease types that I've been interested in up to this point. I haven't looked at aging as a driver, B, a comorbidity, or C, any of these other functions that are driving. I work primarily with neurological diseases, so how the technology that I'm using can be applied to something that affects everyone, rather than, you know, I think about, oh, this affects 700,000 people a year. Well, aging affects.
right, that's right.
Right.
Right, cool, that's great.
I think the surprise for me was, aging and synthetic biology, that sounds like a couple that gets off. Okay. I think a lot of the stuff that, particularly stuff that we've been working on now in synthetic biology, I think it sounds like just based on the last 2 days, that kind of the needs, just making basic regulatory connections. Tool building, which I argue synthetic biology is still very much a tool building. And reporters, and the reporters get come. We didn't talk about it as much today, but I think our field is really kind of poised. We've been, a lot of us focusing on cancer. I've been focusing on cancer. Should being in Houston and all, but I think having aging as a focus for driving a lot of this tool building is I think something that's very real based on this discussion. So
That's right.
it's not like a decades off interaction.
Cool.
Probably right now.
That's cool.
I was gonna say something like that, which is sort of like not only just the opportunity, but the gap is
Right.
far more identified than I think I thought it was going to be at this conversation for some of the fundamental pieces.
Maybe I can say something that impressed me a lot. I didn't realize how stimulating it is to discuss with people who come from a really different field, who have really different, sometimes thought process, or the questions that were asked were like, oh, I have never thought about it that way. And I really enjoyed that. So I thought that was really cool. Thank you, Paul.
To build on your two comments, is I think it's important to recognize that the aging biology field is very, very young. It really is finding its way, and that's where I think there's a model of scientific progression that can be applied. Like, so take your ideas from cancer biology. This is how cancer biology is much older than aging biology by a long shot, and much more advanced, and much more money has been invested. So I think you can use those models and then just start looking for ways of applying them. The other thing I wanted to say is it's always interesting to think who's not in the room, because it's not like this is a very small group of people.
Sure.
Interesting perspective.
Sure.
The people who are not in the room that I've noticed in our conversations are neurobiologists, because I still think that's the #1 challenge in aging moving forward. And I do think because we have no strategies for addressing that, we can get rid of old cells, we can do lots of stuff, we can make muscles do better. That's where I think the true synthetic biology is going to be necessary to solve problems that are otherwise completely biologically inevitable. Because neurons set up and they build their connections, and they just got to live as long as they can. And I think we'll need artificial means of helping them limp along late in life. And I have no suggestion on what that will even look like, but I do think that that's the 50-year kind of goal that we'd be looking at, I think.
Yeah. Well, to that point, and AJ said that too, right? Like the aging is related so closely to many other age-related diseases that what gets developed for aging as a toolbox may actually benefit a lot of other things, which I actually think is really important. Joanna, what do you think?
Well, I mean, I've been amazed to see like what the synthetic biology field can offer. That's a field that I know really little about. I've got an engineer degree in biotechnology, so I got like heard a bit about synthetic biology, but that's not the path I decided to follow on. And so I'm really excited to see how it's possible to connect two fully different fields together, and how they can like synergistically help each other to contribute to like take advantage of like a current tool to learn more about aging, and maybe aging to optimize and fine-tune existing tools. So that's really stimulating to see this happen.
Great, thank you.
Any other thoughts? Can we move on to the next question? Okay, so we're trying to also just capture as much as this for, oh, great.
Sorry, we have a comment.
Yes. Essentially, neurobiology is very challenging. That's interesting.
Why is that? Because of the brain?
Biology lab. And we were supposed to, that's a station.
To the next meeting.
Yeah, that's a great point. It'd be great to hear more about what the challenges are. I'm not sure this is the easiest form. That is a great thread to pull on. To Alexander.
That was all, okay.
Maybe a format. So one thing we do want to do is take a little bit of time, and I'm looking at you, Carmela. And see, we had talked at the beginning of the sort of like launch webinar about the possibility of doing some things in the backside of this. And so just like as a reminder, this is a meeting hosted by NIH, and then there are things that NIH is not really going to be the one to drive. And one of that would be, so there's going to be a meeting report. Sorry, am I stealing from the later things?
That's okay.
Okay, so there's going to be a meeting report that I think Tuba already
Our
told
sons
you,
writers.
right?
okay.
From our
Thank you.
They've been
professional
working hard.
writers in the back here. And the goal of that report is to capture for the most part what's here. If we wanted to, and so that'll be a product of the meeting, and that's part of what you heard about in the intro to this.
Can you just say one more thing about that product? Who are the consumers of it and what is the publication venue for that?
That's right. Okay.
NIBIB website, the consortium website that I put on in my introduction, and it's gonna be for the general public.
In like a documentation of stuff more than, let's say, synthesis into a viewpoint argument. All the opportunity stuff we just did in the last hour and co has kind of a different flavor than some of the things we've been talking about in terms of just documenting the different ideas. So what I wanted to ask, and I'm maybe going to come back because I think Carmela had suggested a potential venue for this. It would be really neat to write this into something that's like a co-written by the folks involved in this discussion the last couple of days, and might synthesize some of these neat insights, including like the surprises question was loaded, because those are oftentimes like most interesting parts when you're coming from a different field, right? So I wonder if that is worth talking a little bit about here as to, is there, you're not committing to being first author if you raise your hand, but like, would people be like potentially interested in contributing to a joint written thing, be short?
Sure.
Okay.
Great.
Okay. Then what we would need to do is decide where to write that as a way of kind of figuring out the dimensions of that. Carmela has suggested ACS synthetic biology as a place that could introduce that.
That's great.
To the, and again, that would shape the audience a little bit, right? But depending on who we're trying to get on the grid, and maybe actually back to your comment about the fact that aging is kind of newer and smaller, might mean that is a good direction to push first, and then we could do another version of that maybe.
I think so. I think that getting synthetic biologists interested.
That's right.
Right.
Getting aging biologists.
I mean, because my take on it is like this will get synthetic biology people interested, and aging folks who aren't already interested will probably be more interested once it's doing something.
Exactly.
Yeah. Okay.
Right,
So.
right, yeah. Right. That's great.
Okay, cool. So that's the thing, and we'll have to figure out some way to do the like mechanically and whatnot. At some point, we need someone who volunteers to lead our process, not to lead the writing per se, but just to lead the cats that need to be herded.
Just to provide a little, I guess, additional helpful details. So ACS synthetic disclosure, I am an associate editor for ACS synthetic biology.
It's already accepted. You
So there are some options, right? So depending on, people's bandwidth, if the idea of like, a 50 reference review doesn't sound so exciting, there are also sort of newer options that aren't integrated into our authorship instructions just yet. Viewpoint viewpoint piece, very short, tight, handful of references. So viewpoint though is meant to be argumentative, like not enough people are doing this right now, and this needs to be done, and everybody wake up. The second option is a perspective, so a bit more toned down, not as opinionated, but just sort of. I think that would be a good fit, right, for some of the things we've discussed. So
Right.
20-something references, not quite as bulky as a review. So sort of.
I like that. Because that could also be a perspective, then something easy for other people to read, right? They don't have to read a 10-page something.
Right.
And perspective still allows opinions but no, I think that's great.
We can write a viewpoint on the perspective and get 2 publications.
Right, that's right.
Yeah. Hey.
Can we do opposing viewpoints on the perspective?
Yeah, that would be actually very interesting. 18 beats in like red, blue. Yeah. Okay, cool. I think that's a great idea. And so
Okay.
did anyone say I'll organize the cats? That's the last business item for today. So
For this paper.
yes, not just for paper. Maybe we have Slack, so we have a thing we can communicate on.
Right.
If you're not on Slack yet, you can access it. That's a place we can kind of like not put people on the spot today. But I'll make a channel that's called Symbio Aging Perspective or something for now.
Okay.
And then let's just like see if we can get things going. And maybe then once we get someone to say they can lead this. And by the way, this can be a trainee, it doesn't have to be API, it could be anybody who's willing to do it. We all have some shared experience on how to do that in mechanics ways. A lot of us do. So I'd be happy to help that person to do to think about what practices might make it easy for team writing. I can't personally give time to doing that right now. I can participate in the writing.
I'll
So-
make one small suggestion is we might, when you set up that channel, people just might write a small, I'm gonna call it a mission statement. It's like, just like, what is this paper gonna be? Because it can be many things, right? So if we could just have like a 1 or 2 sentence statement, and then different people can contribute different things, and that can at least start defining a scope. And obviously we just move quickly to an outline. I think we can pull it together quickly.
We can do that, yeah. And I was gonna say, I think the way we've done it in the past to make it possible to manage people's time burden well is to have just maybe organize a series of Zoom calls where we can do a lot of it in like real time working from basically just exactly what you said.
Right.
And then once you get to that, but just as you said, I would even say sometimes the title drives the mission statement and kind of at the top, and then everything can kind of be fleshed out and delegated from that point.
Yeah.
Cool. Okay. So it'll happen, and timeline wise, it's worth thinking about. It would be useful to have access to the meeting report, I think, to write this perspective, maybe not necessary. And so the thing we learned from our NIH colleagues is that that's probably end of the calendar year, early next year, when that final version of the report will be ready. Is that a pretty good estimate for everything overall? Not the first version of it for
Yeah.
y'all, but like as far as the process.
With all the holidays coming up and, we need some time to go over the report, I would say end of the year earliest.
So one thing to think about is whether we want to make this happen after that, or whether we want to kind of like take the stuff we talked about and use that as material that's adequate for getting things going today. We can do it either way.
I would also say that either sub-channel or whatever is, as people have been pulling up key references or exemplars, be good to load those in while they're still, because I think that will also help us think about, because I imagine our titles are going to be like the emergence of synthetic biology of aging or something like that. The question is how much of it already exists, and that's the review.
Right. It'll be like pack your
Catchy
age
title, but-
or something like
Nice.
catchy, like, yeah. There are a couple channels on here already for resources, and the kind of roughly divided up into symbiote and aging topics. People have been putting them, I think, into places on Slack too, so.
Great.
Great. I think we covered the main for that one. There was one other thing, which is we talked about maybe doing just a survey of some sort for after the fact here, and that could be about like feedback on the workshop. It might actually be also designed to include some things that interrogate your thoughts on some of the scientific content that we talked about here. And so no one wants to do surveys generally, so I wanted to like get the temperament on what the value on people, like they thought like, here's a really good thing I'd like to know, like among the team here. Certainly feel free to elevate that. Otherwise it'll be something that will come from probably Birgit and I, not from NIH, for the reasons that are sort of like, roles and responsibilities. Just so you guys know, that is a real email if it comes, or Slack from us.
Right.
Yeah, are there anything you'd be really curious to know from everybody here? In that, basically, if you'd like to suggest something to go into that, why don't you Slack us instead of saying it right now?
Right.
Something like for email.
Exactly.
And that'll be like probably in a week.
Right.
So it's fresh,
Yeah, yeah.
but not today.
Yeah, so I hope everybody had a bit of fun time, but I think we had a lot of good conversations and we were all pretty active. I was just saying earlier how much nicer it is to actively discuss versus just listen to somebody give a lecture, right? So I feel it was quite inspiring, I think. So yeah, that was cool. And yeah, I think this could have like a big future, and we can tell our friends about it. And I mean, such a perspective will also help in that, I feel. And yeah, and then I mean, the other thing what would be cool is if within your subgroups you come up with some ideas. It might be fun to let us know a little bit, you know, or what was really helpful, or you know, and
Yeah, and I'd say, feel free to also, if you want to use the Slack tool to facilitate any of that, I just made that up. It was not claimed as a team name. So if someone at some point wants to make that into a paid thing that you own, let me know. I'll transfer it over to you. But it's currently, if you haven't used Slack, this is why I'm cautioning. Great place to discuss stuff. At some point, if we don't pay them, some of that becomes inaccessible. You can recover it for money. So we can do that if necessary. I was just saying,
Sure.
use Slack until you want, but maybe this is like think about it for the next, probably a year.
Period.
It's not the free part. It's free right now. It's like up to a certain number of messages. My guesstimation is like, we wouldn't hit that for a
So I
year.
think 90 days and then it wipes things beyond 90.
Oh, that's right. They changed it to
Yeah,
that.
which is really important. And save it.
That's good to know.
I just don't know how long until that starts to get wiped.
I just want people to kind of be aware of this. It's not very expensive. If we were to just like restore it and download it. I actually have an undergrad who wrote a Python script for converting all those JSON files into an interpretable format because they make it very hard to interpret. But so we can capture if we need to. Don't worry too much about that. Just wanted to mention that facet of it. Okay, so I think the next thing that's going to happen is we're going to hand over the reins to Tuba for the client remarks. So before doing that, I just wanted to say, for me, like us, thank you everybody for doing this, and thank you to all of our colleagues from the NIH who made all this happen and made so many great contributions scientifically as well as the organizationally to all the things that we did the last couple of days. So
Yeah.
thanks very much.
Yeah, thank you. Yay.
I have not much more to add. Thank you so much for coming here with all your ideas and participating in the discussions. We really enjoyed it. I learned a lot. Thank you. Hope you make some connections. Keep these connections going. That's the goal of the meeting. And reach out and tell your friends. Next NIH deadline is October 5th. Just kidding. Come back to us with some grants and check out the NOC as I mentioned. If you have any questions, feel free to follow up afterwards and we're happy to guide you. So thank you, everyone.